vascular injury. So vascular injury is nothing but whenever a blood vessel is having some cut in between so that the blood is leaking out. So what is going on? Let us see. The platelets mainly release the serotonin at that time. Whenever there is injury at the site of vessel, there immediately the serotonin get released and it leads to vasoconstriction, means the blood vessel narrowed. So when the blood vessel narrowed, there will be low blood flow. The blood flow will be low. So because of this, we can minimize the loss of blood flow because already there is cut, so that more blood flow will be going. So to prevent this, serotonin is immediately released so that more blood will not be going on. So this will lead to decrease in blood loss. So after the blood has come out, now everything is okay. Now again it should seal it by clotting factors. So the clotting factors which are useful for formation of clot, they mainly decrease in blood loss. So our target whenever there is vascular injury, our main target is to minimize blood loss. So first mechanism in that is production of serotonin, which will lead to vasoconstriction and prevent blood loss. And the second one is clotting factors, mainly they form the clot and prevent the blood loss. And in the case of any injury, the inactivated clotting factors get activated. So in the normal blood, now in our normal blood without any injury, then there will be always inactivated clotting factors. But whenever there is injury, only this inactivated clotting factor get activated by different mechanisms. So let us check out what are those clotting factors and what are these coagulation pathways. So we have clotting factors starting from number 1 to 13. So in that sequence of number 1 to 13, except the 6th clotting factor, we have all the numbers like 1, 2, 3, except 6. We don't have 6 clotting factor. So what are the names of different clotting factors? Let us check out. So the first clotting factor we have is fibrinogen. So the first clotting factor is fibrinogen, and the second clotting factor is called prothrombin, and the third one is called tissue thromboplastin, and the fourth one is calcium, fifth one is proaxalin, and seventh one, as I said, sixth one is not present, seventh one is proconvertin, and eighth one is antihemophilic factor A, ninth one is antihemophilic factor B, which is also called as Christmas factor, and factor 10 is Stuart Prower, and factor 11 is antihemophilic factor C, and factor 12 is Hedgeman factor, factor 13 is fibrin stabilizing factors. So these are all the factors we need to remember, starting from factor number 1 to factor number 13. In between this sequence, only the factor 6 is absent. So that is a major thing we need to remember about this clotting factors. Now let us check out the coagulation pathways. So mainly these clotting factors which we have discussed from the number 1 to 13, these are all involved in this coagulation pathway. There are mainly 3 coagulation pathways. The first one is intrinsic, or also called as contact activation pathway. So for example, in vitro experiments, if we do on a glass slide or in a test tube, the blood which is in contact with the blood test tube and with the glass slide, then it starts coagulating. So that is called as intrinsic or contact activating pathway. And the second type of pathway is extrinsic or tissue factor pathway, and the third type of pathway is common pathway. So let's check out each of this pathway. So first pathway is intrinsic pathway. So intrinsic pathway is mainly having factor 12, and this factor 12 should be activated into factor 12A. A means in activated form in the presence of HMWK, which is a high molecular weight kininogen. So high molecular weight kininogen and also calcarein protein, these are two required for activation of factor 12 to factor 12A. And this factor 12A converts the factor 11A in 11 into 11A. That is, factor 12A activates the 11, and the factor 11A converts the 9 into 9A, that is activated form of 9A, and the factor 9A converts the factor 8 into activated form, that is 8A. So this 8A later enters the common pathway. So this is about intrinsic pathway. So intrinsic pathway is first factor 12 activates 9 activates 8, and 8 activates the common pathway. So we need to remember the order: 12, 11, 9 and 8. So later it enters a common pathway. We'll discuss what is that common pathway also. And next we have another pathway, that is the extrinsic pathway, or also called as tissue factor pathway. So here mainly we have the factor 3, that is a tissue thromboplastin. In the presence of tissue thromboplastin factor 3, the factor 7 is getting activated. So this activated factor 7 this activated factor 7 also ends up in the common pathway. So in extrinsic pathway just have in the presence of factor 3 we will activate the factor 7, only that one, whereas intrinsic pathway first 12 9 12 11 9 8. So finally this extrinsic and intrinsic pathway end up in a common pathway. So what is this common pathway? Here actually the factor 10 gets activated, and this activated factor 10 converts the 5 into activated form, and this 5A converts the prothrombin into thrombin. So along with this activated 5A we also need activated 10A, and also we need the calcium. So in the presence of activated form of factor 10 and factor 5 and the calcium, the prothrombin is converted into thrombin, and this thrombin in turn it converts the fibrinogen into fibrin. So this fibrin is a definitive clot, and the factor 13 again acts upon this fibrin to form a definitive clot. So this is the main formation of clot. So three pathways are very important: extrinsic pathway, intrinsic pathway, common pathway. Extrinsic pathway is simple with a 7 factor, whereas intrinsic pathway have factor 12, factor 11, factor 9, factor 8, and the common pathway have factor 10, factor 5 and factor 13. So these are the factors we need to remember. These are complicated pathways. So finally we'll see how we are going to assess this clotting factors and anticoagulation pathway. So assessment of clotting factors is mainly done by activated partial thromboplastin type. In the short form it's called APTT. This is an assessment mainly for the intrinsic and common pathways, and the normal time limit for this should be 26 to 40 seconds. So if it is within 26 to 40 seconds it is said to be normal. If it is above this or below this, it is abnormal. So to assess the intrinsic and common pathway we need this APTT. And as prothrombin time in the short form it's called PT. So this is mainly for the assessment of extrinsic and common pathway, and here the normal time ranges like between 11 and 16 seconds. So if it is below than this or above than this, then it will be again abnormal. So now we'll see anticoagulant pathway. So till now we understand how the coagulation takes place, what are its pathway, different pathways. Now anticoagulation, because if the coagulation continues for longer time, then it may form some abnormal clots in our body, and this will lead to again some different problems. So to prevent this we also have our anticoagulation pathway, which is lysis, which lyse the clot. So it is now going to break the clot. For example, now we have discussed about the vascular injury. So whenever there is vascular injury, the clot will form and it will seal the blood vessel, and again if the blood vessel become normal, this clot should be lysed immediately, otherwise it will lead to some other problems. So to lyse these clots also we have anticoagulation pathway. So this coagulation pathway and anticoagulation pathway should be always equal and normalized by our healthy body function. So if there is any dysfunction in this, it will lead to different pathologies. So we'll see this anticoagulation pathway. Mainly here we have thrombin. So even if you see, even in a coagulation pathway we have thrombin, but in anticoagulation pathway the thrombin is associated with another substance, that is thrombomodulin. So thrombin in association with thrombomodulin is activating in an anticoagulant pathway, which mainly activates the protein C and protein S. So this protein C and protein S, by this activation they mainly try to inactivate the factors 5 and 8. So when factors 5 and 8 is inactivated, of course the coagulation pathway will inactivate. And also this thrombomodulin and thrombin complex activates the tissue plasminogen activator. So this tissue plasminogen activator converts the plasminogen to plasmin. So plasmin is otherwise also called as fibrinolysin. In the word you can say fibrinolysin. It's lysing, means it's breaking down the fibrin so that clot will be break down. So we need mainly two factors to break this pathway, that is protein C and protein S. Another one is tissue plasminogen activator. So we also have another anticoagulant pathway like antithrombin 3 in association with heparin, which mainly inactivates our factor 9, factor 10, factor 11 and also factor 12. So all these, if 9, 10, 11, 12, all these factors are inactivated, of course the coagulation pathway is inactivated or the clot is lysed, so mainly by antithrombin 3 and heparin. So this combination is important. Before we are seeing thromboxane with thrombomodulin, and now we are seeing antithrombin 3 with heparin. This combination only work better to inactivate these factors. And next we have tissue factor pathway inhibitor, that is also called as TFP1. So this is all about this anticoagulation pathway. Now we will see there is also some regulation of anticoagulation pathway, because even anticoagulation pathway is more like now all the clots are getting lysed, means even at the site of injury, if the clot gets lysed before it is getting sealed, then it will lead to severe blood loss or hemorrhage. So to prevent this also, we should have some regulatory factors like regulation of anticoagulation pathway. It is done by antiplasmin. So in the word you can see antiplasmin means anti-fibrinolysin, means it will not allow fibrinolysis. So this mainly inhibits the plasmin. And also we have plasminogen activator inhibitor. So you should understand here, plasminogen activator inhibitor means this tissue plasminogen activator we discussed already, which converts plasminogen to plasmin. So this tissue plasminogen activator is inhibited. So if you inhibit this, of course there will be no fibrinolysis and the clot will proceed further. So this is all about our quick revision of vascular injury.